I ask every day, anybody have any questions about anything? You ready for some cold weather coming up? Not me either. Okay, today we're going to talk about a cell structure, a set of structures called the cytoskeleton. And this is a relatively new discovery in terms of our understanding of what the cell is like and how it functions. When Dr. Morgan and I were sitting in your seats taking biology classes as an undergraduate, they didn't really know about the cytoskeleton, and there was really nothing in the textbooks about this particular structure. Improvements in microscopy and also improvements in various staining techniques, particularly the use of fluorescent antibodies, really allowed us to detect this intracellular network made up of a bunch of protein tubules of various diameters. And there are three types of elements that make up the cytoskeleton. They are listed in order of their size, decreasing, so microtubules are the largest, then smaller are microfilaments, and then finally the intermediate filaments. What does this cytoskeletal network do? Well, its name gives you somewhat of a hint. One of the big things that it does is it provides intercellular shape and support to the cell, kind of like your internal skeleton does for your body. But in addition to that, it also is involved in cell motility. If cells have the ability to contract, it's involved in that process. And also it's involved in organelle movement. And when we saw that video, The Inner Life of the Cell, one of the pictures that I think is really dramatic is this great big vesicle kind of walking along the surface of this big tubule. That's an element of the cytoskeleton. And I did tell you when we were looking at cells that in reality, there's a lot of stuff happening within the cell. It's very dynamic. And organelles are moving all around. And many times they are kind of moving along the surface of the cytoskeletal elements, almost like kind of a train track that they're motoring over. We're using our white blood cell as an example of kind of a cell to kind of tie together the stuff we're talking about. And in the video, you also saw at the very end that white blood cell undergoing a really dramatic change in shape, where it got really flat and thin and then was squeezing its way out of the circulatory system into the surrounding tissues. If the cytoskeleton is involved in maintaining cell shape, then the organization of that cytoskeleton is going to have to change if the cell changes shape. And those were some of the images that you saw in that video as well that were kind of dramatic. There were these tubules that were growing in length. And then there was tubules that were shrinking. The elements were taken away from them. Those processes are called polymerization. That's when they grow in length. And then those tubules can also depolymerize where units are broken off of them. So the cytoskeleton is a very dynamic structure. So we're going to look at it a little bit, look at some of the elements of the cytoskeleton, and try to give you some kind of an idea of what's going on and how it works and its importance in the cell. So here is an artist's rendition of a cell, showing some of the other organelles that we've already talked about. And then in yellow here, you can see these little kind of gourd-like structures. They are protein filaments. And you can see here that they are pretty extensive throughout the cytoplasm. And in this case, even protrude into these little protrusions that stick out from the cell surface. And cytoskeletal elements inside of those little kind of pockets help to maintain that particular structure and allow that to happen. This gives you an idea of an electron micrograph of some of the cytoskeletal elements. And you can see, for example, that there are some larger diameter ones here, and then some smaller diameter elements over here as well. But those proteins all interact with one another. These things polymerize, depolymerize, join together in different ways. And that has some very profound consequences in terms of cell structure and function. So, three elements. The first one, and the one we'll spend the most time talking about, are the largest elements, the microtubules. They're made up of a protein called tubulin. And tubulin is a polypeptide chain that is wound up in this helical fashion, okay? So each one of these little circles right here represents an individual polypeptide chain made up of many amino acids. And those polypeptide chains begin to organize with one another, forming this helical structure, which in effect gives you a hollow tube. It's almost like the tube that you find inside of a roll of paper towels or toilet paper, okay? That would be analogous to one of those microtubules. These are cells that have been stained by fluorescence microscopy, and the green is specific for antibodies that will bind to this protein tubulin. So you can really get some kind of an idea. Here's an individual cell, here's another cell, here's another one. You can see the nucleus standing out very well here. And then you can see how much of the cytoplasm that these tubular fibers fills up. What do? They help to maintain cell shape. They're involved in cell motility. They are involved in chromosomal movement in cell division. And they are also involved in organelle movement. Let's talk about this one for a second, cell shape and chromosomal movement. Because you talked about that last semester. You talked about mitosis and meiosis. Do you remember what the structure is called to which the chromosomes attach and begin to line themselves up before they ultimately are split apart? It's called the spindle apparatus or the spindle fiber. And what does that consist of? It consists of microtubules. Where do they come from? They come from the cell's cytoskeleton. So prior to cell division, the cell's cytoskeleton undergoes a dramatic reorganization. And a lot of those tubules depolymerize and repolymerize and basically reform into this structure that's going to be involved in segregating chromosomes. Well, if the cytoskeleton is responsible for maintaining cell shape, what should happen to the shape of the cell prior to mitosis and meiosis when the spindle's forming? Got one shape before, which is determined by the organization of the cytoskeleton. Cytoskeleton then reorganizes into a different structure, the spindle. What's going to happen to the shape? It can change, right? And you can actually see some very dramatic changes in cell shape prior to them undergoing mitosis and meiosis because of this reorganization of the spindle fiber. In plant cells, there is a system of microtubules that are organized together to form a structure called the centriole. The centriole helps to organize the microtubules into that spindle apparatus. So it somehow controls that reorganization of the cytoskeleton into that spindle apparatus. Do animal cells have centrioles? They do not. This is actually a plant cell structure. What? Other way around. You're right. I'm sorry. Animal cells have the centrioles. Plant cells do not. Thank you. That's one of the questions about cell structure then is, why is a centriole needed in one type of cell but not another type of a cell? Okay? How can a plant cell organize its spindle apparatus without this structure, the centriole? So that's something of a mystery.
I have a quick question.
Yes?
So you said the microtubules form a helix. Does that mean it has a the cell structure, the coordinating structure?
That's what they would be talking about right here, okay? And so individual proteins aggregate together to form this tubular structure. When we talk about primary, secondary, tertiary, and quaternary, we're really talking about the structure of individual polypeptide chains. So when they're talking about the tubular nature, they're talking about the interaction of all those proteins.
So what structure?
Well, I mean, those terms really don't apply to this helix. It's not a level of organization of a polypeptide chain. It's really kind of almost a level of quaternary structure, I guess you might say, because there's several interacting polypeptide chains to form this complex tubule. Okay? Okay, cilia and flagella. Those of you that have had lab have seen some motile organisms, particularly the paramecium is very motile. So these are structures of cell motility. What's the distinction between a cilia and a flagella? How do you tell the difference between the two? Huh?
Number present.
Number present. Which one are more numerous typically? Cilia are more numerous, and what's something else that separates them from flagella? They actually both move in the same fashion. Cilia tend to be shorter. Okay, flagella tend to be longer. So here are flagellated sperm cells. And here's an electron micrograph of that sperm cell. Basically, the cell body contains a bunch of DNA packaged into it with a whole lot of mitochondria. And then this single flagella, which moves by undulating back and forth. Why is it significant that there are tons of mitochondria packed into this head of the sperm in addition to its DNA? What do they need the mitochondria for?
To fuel the motion, they need more ATP.
Okay, they need ATP for this flagella to move and undulate back and forth again. Ciliar movement also requires ATP. So this is an energy-dependent phenomenon. Now, something I should point out to you is that both prokaryotes and eukaryotes can have flagella. In eukaryotes, the flagella has an arrangement called the 9+2 arrangement, and we'll show you what that looks like in just a second. And the way this flagella moves, is by undulating back and forth from side to side. Okay? So that's how you get your movement. Even though prokaryotes can also have flagella, not all do, but some do, they have a very different structure. I'm not going to get into the structure, but it's not a 9+2 arrangement. It's a very different structure. It's basically one hollow tubule of protein. And they move differently too. The flagella in a bacteria rotates around like this, kind of like the prop on a motor boat engine. So it rotates like a propeller around and around, as opposed to swinging back and forth. So they really have different ways in which they accomplish their similar goal, and that is to be a structure of motility. This shows you a ciliated organism, and you can see, sorry about that, you can see all of the cilia that are protruding from the surface of this individual cell. And again, the motion of the cilia is this side-to-side whipping back and forth motion, which then provides the momentum that pushes things forward. Quick break. The cell organelle consists of two compartments separated by a membrane, which is necessary for chemiosmosis to occur. Which organelle are we talking about? See a couple of correct answers. Or, no? That is characteristic of the mitochondria. Okay? Mitochondria, chemiosmosis is a process whereby we can make ATP using that hydrogen ion gradient. Okay, here is an electron micrograph of a longitudinal section of a flagella or cilia. In this case, it would be probably cilia. Longitudinal section means that we cut something lengthwise and then can look inside of it, as opposed to cross section, meaning we would be cutting it in this plane. So when we see this longitudinal section of this particular cilia right here, the cilia is actually an outgrowth of the cell membrane. So each cilia is surrounded by a piece of the cell's plasma membrane. But then stuffed inside of that are these long tubules of proteins, these microtubules. And they're arranged in this 9+2 wheel and spoke arrangement. Okay, so again, here is the cilia protruding out from the membrane of the cytoplasm, of the plasma membrane. Here in the longitudinal section, you can see these microtubules. And then if we do some cross sections of these, we can see what it looks like in that different view. So, what's the 9+2 arrangement mean? Here we have 9 doublets of microtubules. You can see them better right here. In the center, there are 2 singlets of microtubules. And then what you can't really see in this particular electron micrograph very well are these little red proteins. Those little red proteins are a very important protein called dynein. The reason that these cilia or flagella move side to side is that the position of these little dynein proteins flips, and they move and grab a different part of this tubule as they reorient and reattach to it. And when they flip and reattach to a new part of that, that puts a strain on this particular structure and bends it to one side. And then they flip back again to their original position, so it moves here. And then they flip to another position, and it bends it over this way. And then they relax, and it bends back again to the middle. And so they're moving back and forth, side to side. These whole bundles of proteins are stressing by tension back and forth because these little arms are changing position. They're flipping from one shape to another and grabbing onto those tubules at different points. So why does this require ATP? ATP is responsible for these little dynein proteins flipping and changing shape. Proteins do something very interesting. If you stick another functional group, like a phosphate group, onto a protein, that process is called phosphorylation. It changes the shape of the protein. Phosphorylation changes the three-dimensional shape of the protein. So, the protein might look like this shape initially. But if I then phosphorylate it, if I had a phosphate group onto it, that then causes the shape of the protein to change to something else. If the phosphate group is removed, it goes back to its normal shape. So these proteins can have two different shapes or conformations depending upon whether they are phosphorylated or dephosphorylated. Well, that's what's happening with these little red dynein proteins. When they're phosphorylated, they have one shape. When they're dephosphorylated, they have another shape. And the consequence of that shape change is the movement of this cilia or flagella back and forth, side to side, as we create that tension in those bundles of proteins. That's why we need the ATP. Many times, and we'll talk about this more later, people have this kind of incorrect idea about how ATP allows the cell to do work. And the idea that they have is that somehow it's this magical energy molecule, kind of like gasoline, and that it can be burned and converted into energy to do work. More often than not, ATP does work because when it's broken down, when it's dephosphorylated, it turns into ADP and phosphate. And this phosphate then is available to attach to proteins, and the phosphorylation of the protein is what actually causes something to happen. We're going to see the same thing happen in membrane transport later on. When we phosphorylate a membrane protein, it changes shape and maybe moves something across the membrane. So this is a pretty important concept that we'll come back to and talk about more and more as we talk about cell energetics is, what's the role of this ATP and why is it such an important energy molecule? Well, this is one of the major mechanisms by which it works. Okay, so this again shows you kind of what's going on right here. We've got the interaction taking place between the dynein arms, these red proteins, and the microtubules. And you can see as they are phosphorylated, their position changes, and that then causes a stress in that microtubule bundle, which causes it to bend. So phosphorylation, dephosphorylation ultimately results in this flagellar movement. So we understand a fair amount about how this actually all works. Organelle movement, positions of organelles are not fixed in the cells. That should be obvious by our discussion of the endomembrane system. We talked about how vesicles can pinch off of the endoplasmic reticulum, and then maybe go over to the Golgi apparatus and fuse with those. Vesicles can pinch off of the Golgi apparatus and then maybe move to the plasma membrane. We used to think that they were just kind of floating in this kind of liquid sea of the cytoplasm. But now we know that the cytoskeleton really plays a very important role in how those move. And a phenomena that you saw in the video called dynein walking oftentimes is responsible for those organelles crawling across those microtubule elements. Okay, so here's a motor protein attached to a vesicle, and this motor protein can change shape, it can change conformation as it becomes phosphorylated and dephosphorylated. And so that change in shape basically just causes that motor protein to walk across the surface of that microtubule toward a particular direction. This shows you a vesicle and a fairly significantly sized microtubule in the process of this dynein walking and moving from one location in the cell to another. Intermediate filaments are smaller than microtubules. One of the common type of intermediate filaments are a group of proteins called keratin. Keratins are oftentimes involved in support. They help to maintain cell shape. They help to anchor the nucleus and other certain cell organelles. And we didn't talk about this, but the nucleus of the cell has a very complicated three-dimensional structure itself. The chromosomes are all anchored to the nuclear membrane, forming a complex structure called the nuclear lamina, and the intermediate filaments play a very important role in maintaining that structure. Here's an electron micrograph with fluorescent antibody staining of the intermediate filaments. Notice that these don't form the hollow tubules like the microtubules do. So they're larger in diameter, but they also have a solid core. This is an electron micrograph of a epithelial tissue of your intestine. And so this is the cavity or the lumen of the intestine. Here are these extensions of the cell membranes, the microvilli, whose purpose it is to give you more surface area for absorption. You can see how extensive the cytoskeletal network is inside of these villi. It helps to maintain that shape and structure. And you can see how intermediate filaments are then anchored to even smaller filaments we'll talk about in a second, called the microfilaments. So very complex relationship between all of these protein tubules in the cytoplasm of the cell. Okay, and then finally microfilaments. They're also called actin filaments because the protein subunit of a microfilament is called actin. Where have you heard of actin before? Muscle contraction. What's the other contractile protein besides actin? Myosin. Okay, actin and myosin interact to cause these muscle cells to shorten during contraction. They also help to maintain cell shape. They're also involved in a phenomenon called cytoplasmic streaming. They're also involved in cell motility. There are a number of cells, including white blood cells and the amoeba that you either saw or will see this week, that can move by this amoeboid movement. where they can actually extend their cytoplasm out and then kind of crawl along. These actin filaments are really involved in that process of the change of shape of the cell. And they're also involved in the formation of the cleavage furrow during cell division. I remember what that is. When you get to cytokinesis in cell division, the cytoplasm pinches in two, giving you two cells. That cleavage furrow that forms are these actin proteins that are interacting, much like kind of almost taking a belt and tightening it and tightening and tightening it and squeezing the body or the cytoplasm into two pieces. This shows you the interaction between actin filaments and myosin filaments in muscle cell contraction. They slide past one another. What causes these things to move and change? ATP. And how does ATP do that? By phosphorylating and dephosphorylating these proteins, so the way they interact with one another changes because their shape has changed. This shows you the role that actin filaments play in this amoeboid movement. And as these actin filaments reorganize, the cell membrane changes shape, and then the cell just kind of glides along as that complex interaction continues. Macrophages, which are one of the white blood cells we talked about in the immune system, they're the ones that can eat other cells, phagocytosis, moved by this amoeboid movement. They're constantly patrolling around in your body. And they are looking for things that don't belong. One of the things we'll talk about later this week or early next week is, how does something like a macrophage know that something doesn't belong? What is it that really marks your cells as saying, this cell belongs to this individual, and this cell is an invader? It's very important to be able to distinguish between that because you don't want to attack your own cells. Cytoplasmic streaming is something that, did you look at LADIA last semester, Larry? Sorry about that. LADIA is now illegal in Indiana because it's what, an invasive species? Okay. Well, we used to look at LADIA in the 134 class. It's an aquatic plant and it's only a couple of cell layers thick. The leaves are really, really thin. And if you put the leaf on a microscope, actually, then you can actually see into the cells. And one of the things that's pretty dramatic is all of these chloroplasts will be rotating around the edge of the cytoplasm in a circular fashion. It's called cytoplasmic streaming. And the actin filaments are responsible for that internal cell movement. Okay, let's review, which is true. You need two hands for this one. Okay? All of those statements are true regarding the cytoskeleton. What about dynein walking? The key event in this cellular process. Correct answer is number 4, motility using flagella. Dynein walking refers to that interaction that takes place between the dynein proteins and the microtubules. Cytokinesis and amoeboid movement are involved in the actin part of the cytoskeletal system. And chemiosmosis has nothing to do with this. Okay? Chemiosmosis has to do with formation of ATP using a hydrogen ion gradient. "15 seconds. Stand up." I know that sitting here for a period of time and just listening can get you to lose a little bit of focus and things. I can walk around up here. If you ever find yourself at a point during class where you're kind of losing it and you're doing one of these things like this, do not feel afraid to stand up and move around a little bit. It's not going to bother me at all. Okay? Stretch a little bit. Okay. So, plant cell structures. How do plant cells differ from animal cells? They typically have the same organelles as the animal cells do, with some additions. They have a cell wall. They typically have a large central vacuole. Now, animal cells do have vacuoles, but the plant cell is typically a very large, occupies much of the cytoplasm of the cell structure, and then probably most characteristically, the chloroplasts. So here's our stock picture of a typical plant cell, and you can see the plant cell wall right here. We'll talk about that in a second. Here's our nucleus, endoplasmic reticulum, mitochondria, Golgi. There's several chloroplasts here. And then typically there is this large central vacuole. That particular vacuole is filled with water typically, and things that are water soluble. There are various solutes in there. One of the things that a plant cell will oftentimes do is actively move solutes into the vacuole of the plant cell. Why would they want to do that? We're going to see when we talk next week about cell membranes and osmosis and diffusion that when solutes start to accumulate inside of a compartment, that will have an effect on water movement. And water will flow from outside the cell inside the vacuole if there's a lot of dissolved solutes in there. So what's that going to do to the vacuole? It's going to swell. Okay? And that actually will give the plant cell some rigidity. We call it turgor. You can tell when plant cells' vacuoles are low in water because then the plant tissue loses its rigidity and it becomes wilted. And a leaf that's wilted kind of all folds up. You want that leaf tissue to be nice and turgid and flat so it's got a lot of surface area to absorb sunlight. So the vacuole in some respects almost compensates for the fact that plant cells lack that internal rigid skeleton that gives us support. Instead, their cells are turgid and swollen, and that's what gives much of the support. Well, if the vacuole gets too full, then that's going to create pressure on the cytoplasm of the cell, right? And that's going to create pressure on the membrane of the cell. And eventually the cell will burst, right? What prevents that from happening? Why don't plant cells burst when that vacuole fills up?
The cell wall.
Yeah. The rigid cell wall that surrounds that particular cell basically creates an inward pressure that negates the pressure of the expansion of that vacuole inside. So at some point in time, the vacuole can't fill up anymore. It's kind of like the difference between taking a water balloon and filling it up with a hose until it stretches and bursts. But if you put that water balloon inside of a rigid box, at some point in time you can't pump any more water in because the inward pressure of the box walls will negate more water coming in. Additionally, this vacuole can have some pigments inside of it. And those pigments might be involved in flower color, for example. We're going to do an experiment in a couple of weeks in lab, maybe it's next week, with cell membranes. And we'll use little pieces of beet tissue. And we'll take a little cork bore and make these little pieces of beet tissue that are long, skinny bores of tissue. And then we're going to look at factors that affect the membranes of those beets. And the way we're going to assess whether the membranes have been damaged or not is to see if things start leaking out of those beet tissue segments. What we're looking for is a water-soluble pigment leaking out called anthocyanin, which is a purple pigment that gives beets their purple colors. Sometimes packaged in these vacuoles will be a variety of compounds called alkaloids. Have you ever heard of alkaloids before? Alkaloids are compounds that can be toxic to other organisms. And some of them work in a variety of ways, including being hallucinogens. When animals feed on the plant tissue, if they get sick or high, they might be discouraged from doing that again. Plants can't actively move away from a predator threat. But if they can accumulate some toxic substances inside their cells, and particularly if they have an appearance that the predator learns to recognize, oh, this is a certain shape or color of plant, and when I eat that, I get sick. Therefore, I'm going to stay away from it. You want these compounds to be sickening, but you would prefer them not to kill the predator because dead predators don't learn. Okay? So an adaptation for protection is oftentimes included in these vacuoles as well. On the average, plant cells are a little bit larger typically than animal cells are. What's the problem that plant cells are going to begin to start facing because they're large? Surface area to volume ratio problems, right? If they get too big, then they're going to have a surface area to volume ratio problem. Why can they be bigger than animal cells? In part because of this vacuole. Because the vacuole doesn't contain cytoplasm. Many times this vacuole is so big that it really squishes the cytoplasm into a fairly thin layer right out at the edge of the cell. So even though they're larger and they don't have that much surface area, their cytoplasmic volume has really been decreased by the presence of this large vacuole. So they actually have less cytoplasm than a typical animal cell does. So that's why they can be a little bit bigger. Plant cell wall is an interesting structure. What's it typically made of? It's made of cellulose. Cellulose is a polymer, remember, of what kind of monomer? Glucose. What's it kind of similar to? What's another polymer of glucose that plant cells produce? Starch. What's the difference between starch and cellulose? Has to do with how the polymers are actually bonded together and what kind of bonds are involved. Yeah, there's a kind of bond called an alpha bond and a beta bond, and it does have to do with the orientation of the OH groups, the hydroxyl groups, whether they're up or down. Starch is alpha polymers. Cellulose is made up of beta polymers. Why is that significant nutritionally for humans? We cannot digest them. We can't digest beta polymers. We do not have the beta amylase enzyme. We only have alpha amylase. And so plant material is undigestible, the cell wall is, and it is roughage. It goes through the intestinal tract pretty much undegraded.
Do we still use cellulose though?
There are microorganisms that can break down cellulose that have that beta amylase. And you can find them in the gut of a termite. And you can also find them in the rumen, the extra stomach, of cows. And that's one of the reasons why cows can eat grass and other plants and degrade the cellulose, because they have this other kind of enzyme provided by a bacteria living in their gut. The cell wall structure, so here's one cell and then here's an adjacent cell. The cell wall material is secreted out onto the surface of the cell, and it has a fairly complicated structure. We can just think of it really functionally for us as this is just kind of a whole bunch of cellulose molecules all intertwined with one another. There is a structure between adjacent cells called the middle lamella, which is kind of a sticky substance. It's a carbohydrate, it's a polysaccharide. And one of the important types of polysaccharides in the middle lamella is a substance called pectin. Anybody ever heard of pectin before? In what context?
Jello?
Yeah, when you make jams and jellies. I don't think people do that much more. They tend to do freezer jams and jellies now. But when you would make jar jam and jelly, you would add this thickening agent to it, and that thickening agent was pectin. This is an electron micrograph showing you the central vacuole and its predominance in terms of size within the cytoplasm of the cell. And here's an electron micrograph again. You can see how significant this particular vacuole is and how it tends to kind of squish the cytoplasm out toward the edge of that plant cell. Chloroplast is another very interesting organelle in terms of its relationship of structure to function. I'm really not going to say anything about the chloroplast right now, with just a couple of exceptions, because we're going to get into a detailed look at the anatomy of a chloroplast when we talk about photosynthesis. But a couple of things you should be aware of. Inside of the chloroplast are these series of membranes that are called thylakoid membranes. And attached to these membranes are molecules of chlorophyll. And chlorophyll is the pigment that absorbs the sunlight that makes photosynthesis possible. In photosynthesis, we're converting light energy into chemical energy, including ATP. What's the significance of the fact that these thylakoids are stacked up on top of one another, kind of like coins in a stack? What's that an adaptation for? Increasing surface area, okay? More surface area you have, the more chlorophyll molecules you can have, the more light absorption can take place. These thylakoids are embedded into a fluid-like substance that's kind of analogous to the cytoplasm of the cell, called the stroma. You can kind of see it here if you look at this one closely, but the thylakoid membranes are arranged in such a way that they form a compartment. There is a space inside of each one of these thylakoid membranes. What we have is a compartment separated by a membrane, which then separates another compartment, the stroma. Remember what happens, or what we can do, if we have a membrane separating two compartments. Where do we see that before? Chemiosmosis. We can make ATP using a hydrogen ion gradient. So we'll talk about how that's accomplished in photosynthesis. Once we go through cellular respiration, and you understand how ATP is made during cellular respiration, it's really pretty easy to understand the chloroplast. So we'll come back and talk about chloroplast in a lot more detail. This is an electron micrograph of a portion of a plant cell showing you the chloroplast. Here you can see the thylakoid membranes all stacked together. This clear area down here would be the stroma. Here's a mitochondrion. Here is a peroxisome where some chemical reactions take place involving hydrogen peroxide. Okay? So I've already answered this one, I guess. A cell organelle that contains two compartments separated by a membrane, which is necessary for chemiosmosis. That's true of both the mitochondria and the chloroplast. Okay? Because plant cells have a large central water vacuole, what do they also have to have? The cell wall to counteract the expansion of that water vacuole. These play a role in cytoplasmic streaming, amoeboid movement, and muscle contraction. Those are the microfilaments, especially the actin filaments. Okay, we've got one more cell set of things to talk about, which we won't quite get through today. So we'll pick these up on Thursday again as well. Thursday, we'll also start talking about the next chapter on cell membranes. So if you're in the habit of reading ahead, which I would encourage, I want you to start working on the cell membrane chapter. We will talk about cell surface molecules, the glycocalyx, and then some cellular connections that occur in plant cells and in animal cells. The surface of a cell is pretty complex. This is a cell membrane. You can see that phospholipid bilayer. Here is the cytoplasm of the cell with elements of the cytoskeleton shown here in orange. Here are some membrane proteins, particularly ones called integrins, which your textbook mentioned. And then there's this complicated group of external molecules that are found in the external environment of the cell, many of which interact with the integrins and then, by connection, also the cytoskeleton. What's kind of cool about animal and plant cells is things that happen out here in the external environment can actually have an influence on what happens in the cytoplasm of the cell, including doing things like influencing which genes are expressed in the nucleus of that cell. So a change out here can influence gene expression here. That's pretty cool stuff. One of the most important and common types of proteins that's found in this extracellular fluid is collagen. Collagen is found a lot as a support tissue, ligaments, tendons. It's thought that about 40% of the total protein in the human body is collagen, so it's very, very common. There are other molecules out here in the extracellular surface, some of which are attached to the membrane itself, that are sugar-containing proteins, glycoproteins. And taken together, all of these molecules out here give a cell a unique cell signature, which basically identifies these cells as belonging to tissue of a particular organism and even individual. It's these differing cell signatures on transplanted tissue that causes the phenomena of tissue rejection. Because what's happening? Your immune system is patrolling through your body, checking out these cells, and they recognize this cell signature as belonging. But maybe then when they run into the transplanted tissue, they go, oh, doesn't match. I need to then attack and try to eliminate this tissue. It doesn't belong here. And so that's a real issue with tissue transplantation. What do we try to do to minimize that? We try to do tissue typing so that we find tissue to donate that has a similar cell signature to the host that's receiving it. What's the thing that determines cell signature? Genetics is a big part of it. So there's a much greater chance that if you have a genetic relationship to an individual, you will be a better chance for a donor. But it doesn't always work out. Okay. We will finish up here on Thursday and then move into membranes.